Hi, this is Madeline Kolkol. Today is the 3rd, Tuesday the 3rd, 2019. This is for the Biochemical Testing Lab, Carbohydrates. As you can see, it's been well over 12 to 18 hours since I have completed the experiment, and as per the instructions, it did say that the results would be skewed. What we started out with was kind of a pale yellow concoction, are the solutions with the broth, and now they're a deep yellow color. However, I do want to continue to explain the experiment. So for the S. cerevisiae, let's see, that would be this one, number 1. Here we go here. There we go. S. cerevisiae. The only changes that I note, which is they're not visible right now, though there is some clouding or turbidity in the solutions, actually all of them. They only showed kind of a slight orange discoloration, mostly at the top of the tube. And I didn't see any identifiable bubbles necessarily within the Durham tubes, which was very interesting, and that was within all 3, the glucose, fructose, and mannitol. However, with the S. epidermis, let's see, I think they're kind of visible here. There were bubbles in the glucose and the fructose. The only one that changed with that red coloration indicating that there was no fermentation was in the 3rd, which is the mannitol, S. epidermis, number 3, which was this one. And as the experiment indicated, going past the window of that 12 to 18 hours was going to be too long, and then it would skew the results. So I'm actually, even though this is late, I am happy that I did wait to continue to further evaluate these to see what would happen if the experiment did go on longer than the intended time frame. When I was doing the experiment, one of the things that I thought about was not just figuring out how how their metabolisms may differ and how they might use a different form of energy creation, but was that this biochemical test can kind of help us figure out not just what the microbes do, but where they might be viable. If they're in a place where fermentation would be occurring, or maybe perhaps that's the only way that they can create energy. So not just in identifying microbes, but kind of learning a little bit more about them, I think that it could help with. So I'm sorry for my late one this week. I actually had an eye injury, and that's why I'm in glasses now. I had a corneal ulcer, so I was a little bit concerned that it was not just a bacterial infection, but it was a fungal infection. So I had a white spot on my cornea that actually, when I started thinking about it and I thought about the colonies in our experiments here, I was like, is that an actual colony? But it turns out, I guess it was just scar tissue, and it's since faded. Doctor says that the perforation on my cornea has healed, and I'll be slowly moving back into contacts. But after that scare, I think I might just be using my my glasses for a little bit. So no excuse is a good excuse. They're all excuses, but this is my video for this week. I did find it very interesting, especially because I did wait to do the video. I was able to retain these tubes to figure out what would happen if it went wrong. So that is it. I will see you next week, and have a good day.